I'm Blaisdell, a professor of psychology at UCLA, and I just want to talk a little bit about my friendship and collaboration with Seth and how it has profoundly changed my life and how it's profoundly changed a lot of people's lives. We're going to start, however, with a pigeon. This is the first character of our story, because I'm an animal cognitive scientist. So I study how animals such as pigeons, rats, other animals like that, undergraduates occasionally, try to understand how they think. And it's a tricky business, and we have our tools, our techniques, and our magic. But I was at a conference in Vancouver, Canada, in I believe it was 2002 or 2003, presenting some data about pigeons. Specifically, what I was looking at was how they learned to use a visual landmark, a visual cue as a landmark, to find some hidden location. So one of the things I'm interested in is spatial cognition. And what this shows is a data plot from a single pigeon that has pecked a bunch of times on a touch screen in a little Skinner box. And if it hits the right location on the screen, then it'll get food from a food hopper. And what this is showing you is the density distribution of pecks on the screen. And you can't really see it. There's a little, I believe it's red dot, indicating where the goal would be, the hidden goal, that they had the target they had to hit. And you see that the pecks are where they were pecking on the screen is highly clustered around that location. So they could figure out where the goal was. And there was a landmark on each time they were trying to find this location. There was a landmark somewhere that told them where that target was. And I'm not showing the landmark in the screen, just the data. So they can learn this, and this is after they had acquired or learned the relationship between that spatial cue and the hidden target. Then I put the pigeon on what's called an extinction schedule, where I now still had the landmark present, but anytime they pecked at the target or at virtually anywhere on the screen, they would not get fed. So we called that an extinction trial. And what we see is that from a number of these extinction trials, they didn't peck as much, but the rate of pecking went down, which is typically what you see. But the variation in where they pecked, the distribution of the pecks, enlarged dramatically. And at the time, I didn't know that this was a phenomenon that had already been discovered in the 50s. It was a part of the literature in behavioral psychology that I was unaware of. So I was at this conference in Vancouver, and I had collected these data, and I was still mulling over them, when I noticed that there was a talk by Seth on the program about some rat research that he was conducting. And what he was finding, and it's plotted up here and recorded in that talk, was that if rats were pressing a lever, again, this is a Skinner box, and the rat's in there, there's a lever, and there's a food cup. If they were pressing the lever to get food, that if there would be a cue that would come on, like a tone or a light in the little box. And that cue would tell them that, okay, there's a possibility of food if you press the lever. For one of those cues, it was a high food signal. Every time that signal came on, if they pressed the lever enough times, they would get fed. But there was also another type of signal, a low food signal. On other times in the box, when the low food signal came on, then only about 25% of the time would they get food if they pressed the lever. And the other 75% of the time they pressed the lever, and then the trial would end, the low signal would go off, and no food. So there was a difference there, and in the probability of getting food between these two signals. And if we look at a distribution of the lever press durations, how long the rat held the lever down, and you plot all of the distributions across many hundreds or thousands of trials, what you find is that for the high food signal, that distribution is tighter. There's less variation than in the low food signal. So he was finding something very similar to what I had seen with the extinction procedure with the pigeons. And so I talked to him after his talk, and we went and talked, and this is not a picture of me talking to him after that session. This is actually later on. But nevertheless, I told him about what I was finding. He was like, well, yeah, just because I've been finding this as well, and we're starting to explore this, and said, well, I'd love to explore this in the pigeon some more. And so he said, great, let's team up and do some collaborative work, bring back the pigeons, and continue to look at this these kinds of effects. And so he came down to UCLA, and we sat in my office, and I basically sat next to him while he sat at my computer, and in the space of about 2 or 3 hours, programmed the entire experiment in Visual Basic for us to run the experiment and collect the data to test in pigeons something similar to what he was looking at in rats. And what he's always made the claim in psychology is that while lots of research is focused on learned behavior, rewarded behavior, after the animal has learned what to do, there's much less focus in psychology on variability of behavior. That is, how is behavior variability manifest? How is it modulated by experience? And what's its function? So that was the purpose of our research. And we were going to test Seth's rule, which is not what he called it, what I'm going to call it. And this is from his papers with the rats, that the expectation of reward modulates behavioral variability such that they have a negative relationship. If you have a big known source of food, a nice resource, then you exploit that resource. But when food is scarce, you start branching out and exploring and varying, trying to find new food sources. I mean, this is a fundamentally intuitive idea, but really untouched to a large degree in psychology. So I'm going to try not to go too much into detail with these. I'm going to just give you kind of a gist of what we were doing. This is a study we did with pigeons. Now we took 6 different types of cues or stimuli, and they're just different colors. Here's a picture of a pigeon sitting in a Skinner box with a touch screen. On every trial, we would put one of these colored discs on the screen. The pigeon had to peck the disc a number of times, randomly generated a number of times from one trial to the next, in order to end the trial, make the disc disappear from the screen. Once it disappeared, there was a likelihood that there would be food delivered from this food hopper. If it was a red cue, and they pecked enough to make the disc disappear, they would always get fed. This cue here, I'm red-green colorblind, so I won't say the names of the colors. This cue here, only 35.3% of the trials would end in food. All the way down to this cue over here, where only 0.6% of the trials would end in food. And the nice thing about this procedure, as opposed to the extinction procedure I had, is that with the extinction procedure, subjects eventually just give up, and they quit. And there are multiple reasons why you might get increased variance in an extinction procedure. And whereas here, we're collecting the same number of data for each of behavioral output. For each type of trial, the only thing differs is what food they could expect given the color. What does the data look like? Oh, first a brief little video, just to show you what it looks like when a pigeon's doing one of these. There we go. Knows what to do, but no food on that trial. Little inter-trial interval, kind of like our session breaks. There, next trial about it. Come on, you just gotta hit it. There you go. Get some food. Now what we saw is just plotting the 100% probability signal down to the lowest probability. So just this continuation here is what was plotted on the x-axis of these two curves. What this is showing is the mean location on the screen, the horizontal left, right. You saw that they kind of peck more this way. They don't do a lot of pecking up and down. So most of the variation is actually along the horizontal axis. That mean location of where they pecked on the screen was the same across all the different stimuli types. What differed is, if we look at standard deviation, it increased as a function of the density of reward, or the probability of reward. So that the most likely rewarded stimulus had the pecks had the tightest distribution on the screen. And the lowest probability stimulus had the most widespread distribution. So we've confirmed in the pigeon what he had found in rats. And this was also true in the temporal variation. That is, if a pigeon's pecking at a screen, it's not like it's sort of ballistic, but there's a lot of variability from one peck to the next. Sometimes they peck really quick between, sometimes they peck and there's a little bit longer pause before the next one. You can add up all of these interpeck intervals, and we see that the mean of those interpeck intervals also did not change as a function of probability of reward, but again, a very strong effect on the variation of the interpeck interval. So there's a behavioral manifestation of the expectation of the reward tied to the probability of the reward. I don't want to spend too much more time on nitty-gritty data. I'll just say that we also found this with rats that are running around a board looking for food, and here's a landmark that tells them, okay, this is the one that has food, and the landmarks can be situated in different places. And for one kind of landmark, food's always in the goal cup, and for another type of landmark, it's only there 25% of the time, otherwise the goal cup is empty. And even on these trials, we look at the number of searches before they look in the goal cup, that there's a lot of overlap, but the tail of the distribution for the low probability type of stimulus, the landmark, is drawn out much more. So they're doing a lot more exploring before they finally look to see, is this a trial where there's food here? Maybe. So, and we quantified it in a couple of different measures. Basically, we're seeing the same effect in rats with a more of an open field naturalistic path. And so what do I want to cover here? Okay, so these are both instrumental behaviors. That is, the subject has to make a response in order to get the food outcome. So we wanted to look to see if it was the same in Pavlovian behaviors, which are more of a predictive learning situation. It's basically the same as the instrumental task, except that they don't have to peck at all to get any food. They will automatically start pecking, but they don't have to. And the trial ends in 10 seconds after the cue comes on. And what we found is, even with a Pavlovian situation, now here we kind of did a different way of presenting the distributions, that you see with the 100% trial, there's a spread of the distribution around the center where the landmark or the stimulus is, all the way down to the lowest probability, you get the most variation on the screen. So where they're going on the screen. So we replicated in a purely predictive procedure the effects that we saw from an instrumental one. I'll just skip that for a minute in the interest of time and just say that, okay, so we see that there's a general negative relationship between behavioral variability and reward expectation. That different dimensions of behavior, both spatial and temporal, show the same phenomenon in the same subject, in the same experimental manipulation. That in both pigeons and rats, so two different distantly related vertebrate species, showed this effect. We showed it in both a kind of touchscreen apparatus and an open field. And in both an instrumental learning procedure, which we kind of think of as causal, the animal learns that its behavior causes an outcome, versus predictive, where the animal can learn that a cue predicts an outcome. And we even see it within the trial. I didn't talk about those data, but we see it even within each trial. You can track these effects. So this just reaffirms the idea that variety must be important for new learning. And so coming back to this idea of variation and selection, you vary when you don't know what to do, and you select something, and you exploit when you do know what to do. That we're showing that this is an important thing for the dimension of behavior to study and learning. I recently came across a paper about how this is important in the microbiome, that healthy people tend to have much more rich diversity of species of microflora than do people who are sick. Evolution, in evolutionary biology, they focused on this issue quite heavily. So it's interesting that learning, which is very similar in many ways, learning theorists haven't. And another recent paper I came across, very fascinating, that climate change is looking for heterozygosity, that is a more increased genetic diversity in a population of field surs that are undergoing a big population decline. So the breeding population of these fur seals is declining, and birth weight of the pups that are born is declining. But homozygosity, which is more sameness, the opposite of genetic diversity, So genetic diversity is increasing, genetic sameness is decreasing. So there's with this kind of environment where the natural resources are drying up, the population's being stressed, variation's increasing, which is fascinating. The implication would be that maybe this is a mechanism by which this population can get out of a rut and find an alternative strategy to survive and continue. So Seth opened up new windows in thinking for me, new avenues of research. But it wasn't just the psychology research. It was also what else I learned from him that was so important. So I learned about his Shangri-La diet. I even tried it myself and found it very effective. This idea of Pavlovian conditioning as a regulator of appetite, which he wrote a book and received a lot of attention from. It led me to his blog, which I didn't know about until we started talking about Shangri-La diet. So yeah, you can go look at my blog, we talk about it there. I'm like, oh, I have a blog. And it was the first blog I ever started reading. and so he was writing about omega-3 fats and butter and functions, all this self-experimentation and how it affects his mood and his cognition. And I became fascinated with that. He had an interview with Gary Toms back in about 2007, 2008, when he was doing the circuit for his Good Calories, Bad Calories book. So I picked that up, I was fascinated, I read that, and I became very, opened my eyes, let's put it mildly. Seth also blogged a bit about Weston A. Price, so I got his book and read about it. And it's really opened my thinking into this Paleolithic diet's ancestral health means. And it led me to a whole host of blogs at the time, these are just a few of them, that started really opening my eyes to what's going on in the world and how we're living in evolutionary discordance. So I went on a low-carb diet and cutting out processed foods. Okay. I leaned out, you know the common story, I leaned out, I got rid of GI issues, brain fog went away, I had high energy, my teeth and my gums improved, and my dentist noticed that. Most fascinating to me, a genetic disorder I have called EPP, erythropoietic protoporphyria, so we call it EPP, went into complete remission. This is a disease I have, my mom has, my brother, her dad. I mean, it runs in the family, autosomal dominant. I thought I would have it for life because everybody always does. It makes you unable to go out in the sunlight. You're photosensitive, so you burn easily. Not a sunburn, but a really bad kind of inflammation process in the blood. Now, I can go out in the sun. This disease, since 2009, has been in complete remission. That's what really led me to say, okay, Seth is onto something here. These people are onto something. This is where I met Brent Pottinger, and I also got to know about Nassim Taleb's writings, and I got The Black Swan, I read that, and then it further broadened my ideas, and it connected with the physiology and human evolution and variability, and led us to form the Ancestral Health Society, which was a big success. We have some speakers are here today from that first meeting at UCLA in 2011. Then we moved on to the Harvard Law School in 2012. Seth has been a supporter all through and spoke at the events, attended these events. We brought Nassim in 2013, and that was a big hit. Because the ideas they're all gelling together now. And so we come back to Seth and his personal experience, self-experimentation, N=1. And it's worked for me, it's worked for many of you. But what if we could add them all together? N=1 plus 1 plus 1 plus 1. And it's what expensive human trials try and do. But what if we can start taking our tools in the movements we built together and start crowdsourcing our own N=1 experiments to try and find ways to do our own massive epidemiological studies, as messy as they will be, as they could be illuminating. So my postdoc and I are actually starting up a company called Health Crowds to do this. We'll have an online database where people can put in information about their health and supplements they're taking, lifestyle factors, and we get enough of those together and we can run data analytics on it, and people can find, use data analytic tools to find out any emergent phenomena that come out. Like Seth with his morning faces affecting his mood the next day. Well, if people start doing that and reporting their results in the common repository, is there anything interesting come out of there, and any interactions? So stay tuned for that, still in development, but it's just the kind of platform that continues to exploit the ideas that we're exploring from Seth. So that's what I have to say. I want to say thank you to Seth, a dear friend of mine. It's still a shock to me that he's not here among us anymore, but he's touched every one of our lives so deeply and richly that we all continue to love him to this day. Thank you.